Thanks, Mark. So what I would like to do here is present an overview and highlight first results from a newly SLOAN-funded project on home weatherization effects on the indoor microbiome. So kind of reiterating what Brent and what Jack have said, this is a new project. Take everything with a grain of salt. This is still data and analysis. It's very preliminary. This grant was awarded to us in the fall of last year, and we started actively working on it this February. The U.S. Department of Energy provides home weatherization assistance for energy efficiency purposes for up to 600,000 U.S. citizens, or homes actually, at a funding maximum of $5 billion, according to a law that passed in 2009. And households with incomes at or below 60% of the state median are encouraged to apply for these funds. And both homeowners and renters are eligible to apply. Priority for this funding is given to senior citizens, families with children, and persons with disabilities. And the common procedures to increase energy efficiency in homes within this system is the sealing of building envelopes, and that is done by a weather stripping or closing of cracks and gaps. Further, walls and ceilings are insulated, and often leaky windows are being replaced or sealed. Where deemed necessary, also heating and ventilation improvements are being conducted. At a first installation cost of about $6,500, the annual savings per household amount to about $500. So all these measures aim at decreasing air exchange rates in buildings, and we're really interested in finding out how these decreases in air exchange rates affect the indoor microbiome. So our previous research in the Padgett lab has shown that microorganisms in floor dust are a major indoor source for bacteria. And this is highlighted here on the left hand. Do that over here too. In a unifrac-based principal coordinate analysis, where we compare the microbial communities in floor dust and indoor air, ventilation duct air, as well as outdoor air from HVAC dust. And that HVAC dust is basically a surrogate for outdoor air. And we see that the indoor air and the floor dust cluster closer together and far away from the outdoor air surrogate. We've further shown that the air exchange rate in naturally ventilated classrooms is correlated with the unifrac distance between floor dust and indoor air. So this is shown here, where basically an increase in air exchange rates results in an increase in unifrac distance between the floor dust and the indoor air microbiomes. Meaning, if you have an increased air exchange rates, the communities in the floor dust and the indoor air are more dissimilar from each other. With that being said, the hypothesis for this research project is that an air exchange reduction from weatherization measures will increase indoor associated microbial sources. However, we've heard and we all know that buildings are complex entities and that the story is just not that simple. There are many confounding factors that might also affect microbial communities. Some of them are fixed, such as climates and location. Some of them are fairly fixed, such as building design. And others are more variable, such as cleaning schedules, activity of the occupants, and other factors. So the question really is, what are the factors that affect the origin of airborne communities indoors? And how much does each of these factors or parameters matter? And how large is the influence of the air exchange rate on indoor source-related microbial communities in comparison to any of the other parameters that also matters? With regard to our objectives in the study, we plan to recruit families in 10 case homes and 5 control homes. The 10 case homes undergo weatherization for energy conservancy purposes, and the control homes are being kept as usual. We will determine air exchange rate before and after the weatherization takes place, and measure the microbial communities, both fungal and bacterial, in air and on surfaces before and after those weatherization effects take place. We will support microbial diversity assessments, both for source tracking and to just see what's there, with other environmental data measurements such as CO2 concentrations, radon concentrations, temperature, humidity, and also particle concentrations in the air. Because of the many confounding factors that may affect microbiomes indoors, we decided to start off with a pilot study that addresses the question of what factors matter, like trying to tease out what are the wheels that we can tweak. And for that, we have a case home in Pittsburgh, Pennsylvania, that is a whole house retrofit, and a control in Ithaca in New York, upstate New York, as a control home. The case in Pittsburgh is completely going to be retrofitted with new windows, insulation, and also indoors there's changes that are occurring, tearing down walls and other things that are going on to make that home more to the expected, to what the homeowners now expect. It was built in 1954, and the homeowners now are the second owners of the house. It's a basement house plus a one-story, and it has a vented air heating system. Otherwise, it is naturally ventilated. 2 adults occupy the space, and these adults adhere to a rather spurious cleaning schedule once or twice a month. There's a carpet in that house that is to be replaced in the course of this study. The Ithaca control home is built in 1936. It's a basement plus 2 stories, and it's heated by radiators. Naturally ventilated, it's a naturally ventilated building, and it's occupied by 2 adults and 1 child. They adhere to a very strict weekly cleaning schedule. The flooring is parquet and tile in this place. And one thing I want to mention is we hear about this more and more in studies from our friends in the community, that both places have a dog as a pet. And I think we really ought to pay attention a little bit to the Haleys and the Duffys, and also measure their microbiomes within the house and the indoor microbiome. With regards to sampling and analysis, we're performing a range of measurements, including environmental data measurements, temperature and humidity, CO2 concentrations to infer air exchange rate, radon concentrations to kind of assess the exchange with the outdoor air. For surface samplings, we use swabs and wipes based on previous method development in the Patcher Lab. We use SKC personal exposure monitors, and as well as the liquid impingers for airborne sampling. And we also assess the particle concentrations real time with optical particle counters in 3 size ranges. As for source evaluation, we're taking swabs from the occupants and the pads to actually link the houses to the occupants as well. And we're taking samples from the close by vegetation to track also some of that as an influence. We plan to perform sampling in 4 seasons, where we have done 2 season sampling, winter and spring, as we started in the winter, and we have 1 additional measurement kind of as a baseline. So 3 measurements, 3 sampling campaigns in each place at this point. Quantitatively, we're going to assess bacterial and fungal DNA, targeting universal primers on previous developed methods. And we're working also, again, with our former collaborators on assessing the quantitative DNA targets for plant, human, and canine DNA. With regards to microbial diversity assessment, we will perform barcoded DNA sequencing using the MySeq platform. So I want to focus on in this talk, though, is just kind of highlight, and I think Brent has given a very good introduction to that, how we can use some of the environmental data, our binders full of metadata, if you wish, to already know and gain some knowledge about what is going on with regards to exchange between indoor and outdoor in homes. I will also show some results from the personal exposure monitors at the end. All right, so I mentioned there has been 3 sampling campaigns conducted so far in each place. There were in Pittsburgh in our case home in February, March, and May. and then in Ithaca in March, April, and May. And in Pittsburgh, we had 2 winter conditions, and then 1 spring condition measurement. And then Ithaca, we're dealing with the winter months on the verge of spring measurement. And that is important if you just look at the temperature and the humidity, here shown just as delta T and the delta RH. As we move from winter to spring conditions, we already see that the delta T and the delta RH reduce, and so that kind of tells us already a little bit of what's going on with regards to what is indoors, what is outdoors, and where does it merge a little bit. There we go. All right. With regards to CO2 monitoring, that is a very useful tool, again, as Brent mentioned, to also check some of the occupancy that is going on in the rooms. Again, these are private homes. We can't be there all the time and keep track on what is going on. So we're using that CO2 tracer kind of as a surrogate for human activity in the room. And a typical CO2 trace looks maybe something like that over 24 hours, where you have the CO2 level and ppm over the time, with like moderate activity in the morning, 2 deliberate CO2 releases to then measure the decay, family coming home in the afternoon, CO2 level rising, decay overnight, and then starting again in the morning. At so-called windows of opportunities, we can measure the decay by transforming the CO2 concentrations to its natural log, and then measuring the slope of the linear regression, which gives us the air exchange rate. So this is for number 1 and for number 2. And over a course of sampling, we do that for all the windows of opportunities we create or we have, and average out the air exchange rate. In Pittsburgh, the air exchange rate in the 3 sampling campaigns varied much. In the winter months, there were about one air exchange rate per hour, and they doubled in spring. And here it's important to keep in mind that the air exchange rate here partially results from the heated ventilation system that is on and that is transporting air through the building and like actively pushing the air through the building. And in Ithaca, and this is in May, we see that air exchange rates mainly due to natural ventilation. In Ithaca, the control home has very stable air exchange rates from 0.3 to 0.5 air changes per hour. And there's really not much going on that end, which is also interesting for future analysis as we go through all the data. As I mentioned, we also measured radon in indoor air, and here I want to show how radon measurements can supplement air exchange rate assessments from CO2 monitoring. As Brent said, we have to do some due diligence, and this would be another tool that we could add on to get some more information about what is going on with the indoor system, like seeing it as a system where things are occurring. How do we assess that? How do we get something out of it and relate that to the indoor microbiome? So radon-222 is a several-step removed decay product of naturally occurring uranium-238 in soils, and it has an isotope half-life of 3.8 days. In that time, radon can penetrate the building through cracks and openings, mainly in the basement, and it can only leave the building by exchange with the outdoor air. Other indoor sources for radon include gypsum wallboards, stone countertops, concrete foundations, and if present, natural gas and well water supply. With the indoor sources being limited and a constant radon emission from soils based on the presence of uranium-238, the indoor radon concentrations are mainly determined by the surrounding rock formations. So in fact, the US EPA has issued a map of county-by-county radon zones, dividing into 3 different zones from 1 to 2, based on indoor screening levels, where the highest potential for detrimental health effects is associated with higher levels, above 4 picocurie per liter. And so we see that both of our sampling sites are located in these high indoor screening zones. And we can see that when we actually look at our data. This is radon concentrations in picocurie per liter in the 2 different sites measured in the basement and in the first floor, and the 3 different seasons. And so what we observe here is, first of all, in the Ithaca control home with constant air exchange rates, we see very constant levels of radon in air, both in the basement and in the first floor. In the Pittsburgh chart, we see constant levels in the first 2 months of heating in winter, and then we see a rapid decrease in radon concentrations in indoor air. Giving some credit to my other mentor, Bill, the indoor radon concentrations can be determined from its sources and its removal, where we have an indoor emission rate, as in, and also outdoor air, outdoor radon penetrating the building envelope via the ventilation, removal occurring via ventilation and decay. So at low air exchange rates, at air exchange rate above 0.1, the decay of radon becomes negligible, and also concentrations of radon indoors being much higher than outdoor concentrations. We see that this whole equation simplifies to indoor radon concentrations being equal to the sources, so the emission rate, divided by the ventilation rate. So we can use the radon data to infer something about the ventilation rate in the building. And that is very useful, another very useful tool for assessing the metadata and what is going on with regards to indoor-outdoor sources. And this becomes more clear, I think, if we look on the hourly data, where we have the 3 different sampling campaigns in Ithaca on the first floor radon concentrations. We see that there's a lot of variation over the time of sampling, and that at basically at low concentrations of radon, we probably have a high air exchange rate in the entire building, and at high concentrations, a low air exchange rate. So that is something else where radon can provide a measure for air exchange with the outdoors, and that tells us something about the sources as well. So I just really briefly want to highlight particle concentrations and micrograms per cubic meter, PM10 particles with an aerodynamic diameter smaller or equal 10 micrometers in size. These are our different campaigns, and the numbers here are the mean concentration from replicate measurements with the PM10 samplers. What really sticks out is the high concentrations in May, and what we have going on here is construction in the building. And the question is really, are we also going to see an increase in fungal and bacterial communities in this case? And if so, is that maybe also a source for microbes? And then even without that, just looking at the concentrations in Ithaca in Pittsburgh, they're at least, in Pittsburgh, they're at least 3 times increased over the Ithaca control case. And one of the questions would be also, why is that? And what can we just learn from looking at the metadata about what is going on here? So one of the suspicions we have is that the carpet that is in the building in Pittsburgh is a high source of particulate matter, and it's been taken out in that time frame. And if we look at the data from the optical particle counter, here in March the carpet is still present, and in May it's not. We see that when occupancy goes on, people after the night enter the main building part, the living room, we see the sharp increase in particulate numbers of a larger size range. And we don't see that anymore at the later point in May, where the carpet has been removed. So potentially the carpet is a source of at least particulate matter, and we'll see if we can confirm that with the microbial diversity measurements as well. Another interesting aspect here are the spikes during the decay period at night, particle concentrations. And we suspect that this is from the ventilation air duct that kicks on at night to heat the building. And we're going to investigate whether we see duct microbiomes in our indoor air samples as well. All right, I think I'm just going to highlight briefly that the concentration here in the Ithaca-controlled case overall are just not as high as in the Pittsburgh case, and we could just confirm that potentially that's the carpet is the source. All right, this is my last slide, I believe. Fungal genome concentrations and the particle PM10 samplers, and the different sampling periods. We see for fungi that there's an increase in fungal genomes in the May period, in the spring period, and there's a question whether that is due to the construction going on or just to more air penetrating the building from the outdoors. And hopefully the DNA sequencing data will inform us about that. And that's where it really becomes a powerful tool to combine these two different types of assessments. As for summary, we'll pay close attention to participant selection for our rollout and also to the time of measurement. Probably going to look for active participants that we suspect a lot of dust, so we actually meet our detection limits. Pay attention to seasonal differences. A good measurement time would probably be fall. Maybe exclude houses that have a vented heat system, as it also adds a third component that we need to control for. Pets are important, carpet, I mentioned that. And again, like combining these two methods or method approaches from different areas, shown again that this is a very powerful tool to tell a whole story. I think we ought to all work on this together. I'm going to jump that in the interest of time. But I do want to mention that I have a poster, and you'll sort of come and see it, on my old SLOAN work that is done with Jordan Patchak. And it's about how different parameters affect the indoor air to flow dust unifrac distances. And with that, I'd like to thank you for your attention. And
It seemed like the only thing you had in common at all with the two houses was the dog. So I'm not exactly sure how you're going to make some really strong generalities. I mean, you might use that to your advantage to have all these houses are completely different, cleans are everything, but there's one thing that is the same between them. I mean, is that the plan?
What is that?
I mean, it's a crazy number of variables.
That's right, that's right. What we're particularly interested in is really seeing how we can use some of the environmental data to infer what is important and what is not, in combination with the microbial diversity assessments. So I think with the combination of the two, we can actually tease out a lot of these factors. And the measurement over time will also tell some of what is important and what can we neglect.
The assumption that you're making with your exchange rate is that the space is fully mixed, and in particular with these older houses that are naturally ventilated, that's really a big assumption. Basements have their own exchange rate that can differ considerably, so using radon has got to be checked to see if it's working.
Oh, I absolutely agree, and I think all the building scientists in the room prefer that. So,
all three of them.
yeah. So we're hoping that to actually use some of the optical particle data as well to infer about the air exchange rate. That also in combination with the CO2 concentrations and testing how well the room is mixed by installing CO2 monitors at different places in the room. That is obviously something that needs to be checked. I absolutely agree.
I'll ask.
Again later. Okay. Well, thank you so much.